assay. So we start with restriction enzymes, what they are, what do we use it for. Just give you a little bit of background, the history, definition. We'll go quickly, really mainly looking at their classification, how do they cut and how do we apply. Many of these amplification as well as restriction, digestion, and sequencing, we'll look at it again, starting from this afternoon. And once we get into application, how do we use them for various problems? On Wednesday, I'll share with you on Clostridium difficile, molecular epidemiology of difficile, how we use the various methods. On MDR, salmonella, as well as MRSA, mainly in animals. But just let's continue with the basics. So, restriction enzymes. I hope many of you already know, and you raised some hands. So we have known about these enzymes starting from more than 30 years ago. But what are they? These are enzymes that restrict access of viruses, bacteriophages. Bacteriophages are viruses that infect bacteria. So bacteria developed a mechanism to defend itself by restricting access of these viruses infecting them. That's why we call them restriction enzymes. Everybody's clear about what bacteriophages are? These are viruses. In fact, as a tool also we use them for transduction purposes and others. But these are viruses that infect bacteria. Okay? So bacteria, they are always smart, just as they defend against antibiotics, defend against different things, they defend themselves against the viral growth by restricting access. They develop these enzymes to chop down the bacteriophage viruses. So these are endonucleases that cut or chop the bacteriophage DNA. Now we use it as a tool. Daniel Nathans and his team in 1978, they won a Nobel Prize for medicine for the discovery of these restriction endonucleases, because bacteria produce it, we human beings, scientists came up to use them as a tool. Okay, these are very nice enzymes, endonuclease, not just they cut DNA randomly, but they restrict or they digest specific sequences at a targeted location. Therefore, it really provided a tool, a molecular tool for the use of epidemiology or various, either it's genotyping, cloning, whether you study pathogenesis and others. This really led to the revolutionized molecular technologies, the ability for recombinant DNA technology, particularly cloning of specific gene. The use of restriction enzymes is really a key tool for cloning and other recombinant DNA purposes. So, what are these restriction enzymes or restriction endonucleases? These are enzymes that have the ability to recognize, that is the key thing, recognize targeted specific short sequences, and they cut at that stage, not just randomly shearing the DNA. Okay, so you can really target, as scientists, we can use them for targeted purpose. Here is an example. I wish I had my PowerPoint. I had a more animated version of how they cut, but here is an example of CTTAAG, one of the enzymes we commonly use, many of us, EcoR1. It recognizes the 6 base pairs. Okay, CTAAG, and I'll show you this. So these restriction enzymes, they bind and cut double-stranded DNA. Okay, when we say denature, remember double-stranded is separated into single-stranded. When we say cut, it's the double-stranded is cut in the middle, separated into 2 double strands, right? Okay. So at specific, we call it restriction site or recognition sequence. In this case, CTTAAG or GAATTC is the other way. Okay. So we will look at more details of this. Okay. So these enzymes typically appear in almost any gene or chromosome, or this, I'm sorry, not the enzymes, but these restriction sites. Just it is random occurrence of these recognition sites, right? I mean, all genome is composed of these 4 bases in various sequence orders, right? So depending on whatever that recognition or restriction site is, there is a chance that that sequence is present in some or many locations. And we'll look at the frequency, what is the recognition, how long they recognize, and all those details. These sequences can vary depending on that enzyme. There are so many different types of restriction endonuclease enzymes. Each of these enzymes recognize different lengths and base composition of these short recognition sequences, as short as 4 nucleotides. Like, let's say, MSE1, that one we commonly use. It recognizes AATT. It's a short sequence. Whenever it finds AATT in reverse order, it will be AATT. That is a MSE1 enzyme. We use it on amplified fragment length polymorphism. It cuts in that, whenever it finds it cuts at that location. Some of them recognize very long ones, up to 20 nucleotides, very long ones. Okay? Some recognize short, some recognize long. The cleavage, how they cut, it could be symmetrical, it can be asymmetrical. Many of them cut asymmetrically, like I just showed you here, MSE1. Some of them cut right symmetrically in the middle. Okay, but we'll look at those more in detail. And one key thing, there are different types of restriction enzymes. I'm not going to detail of type 1, type 2, type 3, but there are specific types that are very useful as a tool. Is a type 2 one. Typically, they recognize palindromic sequences. They read the same in both directions. Like if you look at the recognition site for MSE1, on the 5' to 3', AATT, if you look at the reverse weight, it's the same, AATT, right? So we'll look at more examples, plenty of examples. If you look at EcoR1, I mentioned earlier, one of the most commonly used one. The palindrome, look at this. It is from 5' to 3', GAATTC. If you go the other direction, it is the same, right? GAATTC. That's called palindrome. So type 2 enzymes, the most useful ones, they cut within the recognition sequence, like type 1, by the way. Some other enzymes recognize that certain location, but they cut not inside that location. They cut maybe 100 base pair far from it. Not so useful for molecular biology as a tool. Okay? But type 2, they are palindromic, usually short sequences. It can vary from 4 to 20 base pairs, and they cut within that sequence, like EcoR1 cuts on this. Clear so far? Feel free. Not clear? Which way? Which direction? I don't know. Okay. How are they named? Again, this is because of PowerPoint. I apologize. There are some overlapped text, but EcoR1. This enzyme is originated from Escherichia coli, okay, from that bacteria, E. coli. R is the strain ID, RY13 was the strain name where this enzyme was first recognized, and 1 is it was the first identified. From the same strain, you can have 2, 3, 4, others. But that is how usually they're named. The genus, the species, the strain, and then whichever the order of the enzyme identified.